last time we left off talking about DNA supercoiling and looking at why DNA supercoils. Why did we say it supercoils?
It keeps it from degrading. Yeah.
To condense itself. Okay, so what are some of those properties of condensed DNA? Its ability to fit into tighter spaces, okay, and potentially less strain, right, depending upon the environment that it's in. DNA topoisomers. These are just DNA strands with different topology. Okay, different topology. This is a right-hand supercoil and a left-hand supercoil. And that's essentially, if you were to take that in your hand, that circle, and you were to twist it like this versus grabbing it and twisting it the opposite way. Okay? So either a right-hand twist or either a left-hand twist. Okay? So topoisomerases are enzymes that will actually do this. How do these differ from DNA ligases? Well, DNA ligases actually covalently bind two strands together, DNA strands. Okay, so ligase will bind together DNA strands. Topoisomerases will regulate superhelicity. Okay, topoisomerases regulate superhelicity. Can these be separated? And the answer to that is yes. You can do DNA resolution by gel electrophoresis. And the relaxed DNA is that circular one that's up there at the top. Okay? And then the supercoils are down here. And you can see that highly supercoiled DNA and relaxed DNA travel differently by gel electrophoresis. Now, electrophoresis is separating things by what?
Number of base pairs.
Well, it's fast
Well,
as
yeah.
confused.
Because shorter fragments move farther. Right? I
Okay.
always get that back.
Is it
But
that?
there is a size exclusion technique, right? I think there are size exclusion gels that can do that based on length. But this one is talking about separation based on charge, okay? And so the supercoiled DNA, the more compact ones, will apparently travel faster on gel migrations. Now, I didn't quite understand that, but apparently they do, okay? So you can separate the topoisomers by gel electrophoresis. How many people have done electrophoresis? And if you have, what have you done it on?
TAG, I don't remember what it stands
I don't remember either. You think
Okay.
we would know, but
for.
you
It's
know.
something
Caleb?
enzyme, but
I just did it on some type of gel, but like the actual DNA, I did it on two different plasmids, one from PUC8, which I think goes like ampicillin resistance.
Okay,
And then one for MRFP, which is like a red fluorescent protein.
oh, okay, and they were both plasmids?
Yeah, yeah.
Okay.
I actually had to restriction digest them, and I ligated those two pieces together to make a new one.
Oh, cool,
Yeah, I'm doing
okay.
that in cell right now. I actually have a phoresis sheet right now that I just printed off like last week.
Oh, very nice, so you did it in cell this last week?
Yep.
Okay, and so you took two different plasmids, which are the circular pieces of DNA.
Yep.
You digested them, you broke them apart, and then you used DNA ligase to
Fuse them back together.
fuse them back together, and then what else?
Well, and then actually, I grew E. coli from.
Okay,
Yeah, yeah. That
Okay.
was you?
and then what did you use the electrophoresis for?
Well, the electrophoresis, we're using it to measure the sizes of each of the plasmids, but we're also doing it to measure the ligations to see if it makes sense, like given the base pairs.
Okay.
Whether or not we actually got them to be successful. So there should be a set amount of sort of mass based on the base pairs. You can measure with electrophoresis that if you got the ligations right, it'll match up.
Oh, very nice.
Yeah.
That's great. Glad you guys are using that right now. I am assuming we will be using that technique closer to the end of the semester, maybe? Okay. Any questions? Okay. Single-stranded DNA and RNA. These are not in distinct structural formation. They are randomized coils, okay? And there's flexibility and rotation associated with these, so there's no real specific structure. Where do we find like single-stranded RNA? Messenger RNA, where else? Inside of a ribosome. What about viruses? Uh-huh, yeah, you find them in viruses too. Okay, yeah, what else? Okay, what about tRNA? That too, okay. So we'll talk a little bit more about these. So there's also non-self complementary single strands that exist under native conditions. The bases can stack into a helix but no hydrogen bonding. And then you form these hairpin loops that have these self-complementary sequences. And I remember looking and studying at hairpin loops when I was an undergrad in biochem lab. Oh, I'll go back if anybody wants to write any of that down. So that's really just it folding back on itself to make complementary pairing. Riley, did you have?
Well, did you post this slide?
I have not posted it yet because I keep altering these slides. I'm actually cutting slides out to make it shorter for you guys.
Can you please do this before, like, post them before this weekend?
Yeah, absolutely, absolutely. We'll be done with it today. So
Okay.
I can post these today. Yep. So tRNA or transfer RNA, you can see how it has gone back on itself, and it's made this clover leaf-like structure. What does transfer RNA do? What's it involved with? It's involved with transcription or translation. Which one is it?
Translation.
It's involved in translation, okay? And what does it carry with it? Amino acids? Anticodons, okay? And anticodons pair up with three codons in the reading frame. That's right, we're gonna see that in just a minute, okay? I didn't know that they had a crystal structure of tRNA. This is actually pretty cool. And I really didn't know that tRNA had such tertiary structure involved with it, okay? It's pretty crazy looking, okay? I mean, if you go from this slide to this slide, it looks very different, doesn't it? Yeah, it looks very different, okay? So you have an acceptor stem on one end and the anticodon stem on the other. The anticodon stem will bond with the codon or at the ribosome. And this is, I think I got my slides out of order. That's okay. We'll get on with translation in just a minute, okay? Base pairing, there's some alternative base pairing that I actually was not aware of. This is called Hogstein base pairing, and it is different from Watson and Crick base pairing. And as a result, there's some of these weird, like triplex structures that can form as a result of some of these odd base pairings. And little did I realize that some of these are stable, depending upon the environment that they're in. And you know, just stuff that I've been very much unaware of. Kind of wild. So you might have any questions about the difference between the Watson and Crick pairing and the Hogstein base pairing? It's simply just hydrogen bonding to a different nitrogen on the ring. You see it? Yep.
I have a question.
Yep.
Do we need to know like the exact where on the like all of the different nucleotides, like
Where the hydrogen bonding occurs, I would like for you to. I would. So if I gave you two, I would like for you to be able to draw the lines between them where they might be. Okay? Or to at least to recognize that. Okay? Yeah? So also just be able to know which base pairs have 2 hydrogen bonding interactions and which base pairs have 3 hydrogen bonding interactions. Okay?
Adenine and guanine have 2, right? And then, no, I have that backwards. Adenine, thymine.
Adenine, thymine have 2. Yep, it's right there. Yep. Yep.
I mean, I remember it wasn't.
There's also these crazy G quadruplexes. I have also never seen these either. Okay? These are made of 4 guanine bases, all hydrogen bonded to one another in a planar structure. These were actually discovered in 1962 by David Davies, and he proposed that these actually were naturally occurring. And it wasn't until later did we find out that these are actually, they are naturally occurring and found in, even in oncogenes and some other, Riley?
Do the G tails form this? In DNA, a lot of them are ending in G's because of the transcription.
I don't know that. There's one spot they talked about. They exist in transcriptional control sites, promoters, genes, including the oncogene CMYC, and there's also, it says there's also drugs that are targeting these structures too. So I think that they are selective. And as a result of their selectivity, that makes them nice drug candidates or drug targets. Because if there's only a cancer gene associated with that G quadruplex, why not target the quadruplex? Because it's something unique, right, to that environment. Yep. These are actually ligands. These are ligands that can host metal atoms in their centers. So organometallic chemistry is another subdiscipline of chemistry. Has anybody ever taught organometallics here on this campus? No? Okay. So organometallic chemistry deals a lot with these types of substructures where they have metals bound to the middle of these, and many of these are used as catalysts in chemistry. And you think about chlorophyll. Chlorophyll is one of those organometallic complexes.
That has, oh goodness, what is it in there? Magnesium?
Is it magnesium
Yeah, I
that's
think
in the
so.
center of
I remember it
chlorophyll?
from botany.
I couldn't remember if it was. Yeah.
But, DNA also uses magnesium.
Well, why do you think chlorophyll would use a metal in the middle of it? What do we think of metals?
Like conductors of electrons.
I was
Yeah.
gonna say, doesn't it typically have a charge associated with it?
Yeah, it's got a charge associated with it, and they're good conductors of electrons. That's right. Okay. We actually consider and think about metals as being seas or oceans of electrons that can shift very readily, like currents or waves. Yeah.
We had an idea of how enough botany classes here if you learn that in botany.
Oh.
I remember she put up the structure of it on her slideshow. She's like, you guys don't have to memorize this, but here's a little bit about it, and it'll just.
We have to know the magnesium part.
Oh, yeah, we
It's a test
did.
question.
Okay.
Cool.
Well, I got the test question right clearly, but.
Very cool. Here's a side view of the G quadruplex. Again, very, very interesting. Looking at these structures. And there is the N view, which is like what you'd be looking at here, except for having the phosphate backbone surrounding it. So the phosphate backbone is still hanging out all the way around that. It's pretty impressive, pretty cool looking. Okay, let's switch gears. How is DNA destroyed, and can it be repaired?
High levels of radiation can destroy it.
Okay, high levels of radiation can destroy it. Something even more simpler can destroy it. Heat, right? Yeah, heat. Having a fever, okay? So native DNA, when you heat it up, can become denatured DNA, which is, again, single-stranded and it breaks apart. But if you cool it down slowly, and the pairs are still close to one another, they can come back together. Okay, I didn't know this. Okay, that's kind of wild. I never thought that DNA denaturation or denaturing was reversible. But if we think about nature, it kind of makes sense that there should be some type of a repair mechanism to help out with that. Now, depending upon the temperature, it might be too far gone, right? And that's how organisms can die. So as the temperature increases, when we think about thermodynamics and Gibbs free energy, delta H or delta G equals what? Delta G.
H minus.
Delta H
TS.
minus T delta S. That's right. Delta H minus T delta S. So as the temperature increases, the negative T delta S overcomes the positive delta H, which is enthalpic conditions, and then makes delta G negative, which is spontaneous, right? And then the denaturation is favorable, and therefore you get melting of DNA right there at that middle point. What's really fascinating is that you can watch this happen by UV vis. And I think that would also be a really cool thing to do, would be to extract your DNA, take it out, put it into a cuvette, set it out, heat it up, put it in a UV vis, heat it up some more, put it back in the UV vis, and observe this and watch this happen. I think that would be a great experiment to do. Has anybody ever done that before? No? So the absorption actually is less for native DNA than the denatured DNA, but you could literally graph this over time and watch this particular event take place. Any questions on that? Okay. So it happens pretty sharp, and I didn't realize that it happened actually quite so sharp. I would have guessed it may have been more gradual. The book actually talks about it being compared to melting of water or of ice into water and then going back through the freezing point, that it's literally that well defined. DNA polymerase. DNA polymerase is an enzyme that does exactly what it says it does. What's that do? That's right. It polymerizes or synthesizes DNA, right? And so you get the triphosphate, the nucleotide that adds in, gets rid of the inorganic phosphate, and then prepares the strand for the next base pair as it goes. Yep, Riley?
It has to bind something to connect to the strand. As a result, you get overhangs.
Yeah, and what happens?
You need DNA telomerase comes across and adds extra DNA strands. You're not actually putting DNA.
And we watched that in the video, right?
Yeah.
We watched that happen real time in the video that we watched. Yep. So here's an example or a graphic description of transcription. So we have RNA polymerase, and this is transcribing the DNA. And why does it do this? Why does it do this? Okay. Yep. So this is the whole process of getting all the way down to the gene level. Yep. So here's translation, and we've got tRNA with its anticodon. We have messenger RNA that's bound into the ribosome. Most of you should already know that at the ribosome is the site where protein synthesis begins. Okay, so you've got codons that are coding for what? What are they coding for? Amino acid sequences. That's right. Okay, and they just keep building and building and building and building until you get whatever protein that you are essentially synthesizing. Yep. So the free amino acids bind and get translated and keep building the peptide or protein from that sequence. What an amazing machine! Awesome machinery, doing some really cool work in us every day, every second of the day. All right, so here's the whole picture: DNA replication, transcription, RNA translation, all the way down to the protein. Okay? So when we get into the next chapter we are starting at the amino acid level, and then we are going to be moving from there, looking at how proteins are synthesized. These are some images of proteins taken not by X-ray crystallography, but by a technique called cryo-electron microscopy, and this was in your book. These are complex protein structures. And the way they do this is they put the protein in liquid nitrogen and then basically shoot a beam of electrons through it to look at these and to gain images of these really complex structures that you would not be able to gather from a crystal structure. Okay, this technique's actually even been able to improve upon prior X-ray crystals of protein structures that weren't quite as accurate. Okay, so this is a relatively new technique, and I've never seen this technique before. But again, X-ray crystallography is limited simply due to the size and complexity of various protein structures. Even NMR is being utilized for trying to unveil protein and peptide structures. Doesn't it
That
sound
sounds
awful?
awful.
It does, right?
You
Do you
need a
think?
really high resolution MR.
Oh yeah, very, very high resolution
Way
NMR.
too much time.
So the one that we had at my postdoctoral institution, which was Sanford Burnham Medical Research Institute, that NMR was probably about 6 feet wide and 10 feet tall, and we went up it with a spiral staircase to drop the tube at the top. And the room had to be this big in order to have enough buffering space because of the superconducting magnet, and to make sure there wasn't any metal anywhere around that.
Isn't your growing up?
It is, yeah, it is. Yeah. Do you know what it does? Tell me what it does.
When it's triggered by stress in the body, usually heat, and it tries to preserve proteins when like it starts to denature and it brings them into their non-polar regions. When they start to denature and it pulls them on the inside, so it doesn't lose its structure.
That's really awesome. So it's a protein that preserves proteins. Protein that preserves proteins.
It's
Very
more
cool.
stable because of a bunch of disulfide bonds, I think.
Oh, okay. I wonder if the disulfide bonds, are they located on the inside? It looks like this thing. It looks like this thing's almost like a barrel.
It is. It's double-sided, so it can hold 2 proteins, one on each side.
Cool. Very cool. Nice. All right, that's it for this chapter. What's next? What time is it? How are we doing time?
2:32.
2:32. All right, break off into your groups.
You still have the video?
Yeah, you can stop that.